Alright, ladies and gentlemen, this is going to be our next video in biology, and specifically going through and discussing transcription. Let's go through and get started on this next video. So if we're going through and looking at the overview here again, we are going through and looking at the top half of this process here, where we're going through from DNA into our RNA. Now the definition again for transcription is the process of taking DNA and making RNA molecules inside the nucleus to be used later by the ribosome to make proteins. Now remember, we have an enzyme, RNA polymerase, and we have DNA, and we have our mRNA. These are the players associated with transcription. Now transcription is going to consist of three basic steps. The first step is going to be initiation. Second step is going to be elongation, and then lastly we're going to have our termination step. So if we're breaking this down, we are breaking it into three steps: initiation, elongation, and termination. Now if we're thinking about where this takes place, well, transcription is going to go through and take place inside the nucleus. So it's taking place inside the nucleus, inside our eukaryotic cells. Now let's go through and break this down into our steps here. The first step here is initiation, and this is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind, so the enzyme can then read the bases in one of the DNA strands. So RNA polymerase is essentially going through and binding to our promoter region, which is in front of the gene. Now remember, that gene is what's going through and coding for the actual sequence, that actual protein sequence that gives the solid structure in our body. So that gene is the region of DNA coding for the proteins. So step number one, we have this promoter sequence in the beginning here. And our RNA polymerase is going to go through and bind to that promoter sequence just in front of the gene. All right, now let's go on to step two. This is the middle step here. Elongation is the addition of nucleotides to the mRNA strand. So RNA polymerase reads the unwound DNA strand and builds the mRNA molecule using complementary base pairs. So we're going from RNA polymerase here. We're going to unwind and synthesize the actual mRNA molecule from the gene, or the region in the DNA which our protein is going through and being coded for. So RNA polymerase is going to go through and unwind that DNA. What it's going to do is it's going to assemble that RNA sequence there. And specifically, we're going to be going through and getting our mRNA sequence, which we're going to later use in the processes of translation. Now the last step here is going to be the termination, or the end sequence of that step. Termination is the ending of transcription and occurs when RNA polymerase crosses a stop termination sequence along the end of the gene. So at the end of the gene, what we're going to see is RNA polymerase is going to go through and decouple, or it's going to leave, and the DNA is going to rewind. So at the end of our sequence here, our RNA is going to go through. It's going to hit a stop sequence or a termination sequence. Our DNA is then going to go through and rewind, and we're going to be left with, again, our mRNA that we're going to go through and use later in the process of translation. So what is our end result of this process? Well, the product of transcription is going to be our mRNA, our messenger RNA. Now, messenger RNA is a single-stranded piece of genetic material, and it's going to be incredibly important when we go through and put it into translation. So the product of mRNA is going to be sent to the ribosomes, which are either going to reside in the endoplasmic reticulum or the cytoplasm. So just to be 100% clear here, we're going from DNA to mRNA when going through and discussing this process. Alright, so did you learn? Well, did you learn a couple of things here? Did you learn about what transcription is? Did you learn about the three steps of transcription? And lastly here, did you learn about the end result of transcription and what that end result is? This is going to be the end of the video, but we'll see you all in class tomorrow.